Thank you. It's a pleasure to be here. Today, I think I'm going to tell you about something that's quite new in my laboratory, which is to really look at how to manipulate neural circuits to ameliorate Alzheimer's disease pathology. This is a very active area of research now in my lab, and also we are recruiting more and more collaborators to work together. So we now call this system gamma intrument using sensory stimulus or GENUS. So I'm going to refer to this term in my talk. So first of all, why neural circuits? So we know that there are many neurons and other supporting cell types in our brain, probably billions of neurons. And in the cerebral cortex, it is estimated that 20% of the neurons are actually inhibitory interneurons. So these excitatory and inhibitory neurons form trillions of synapses, and they form these very intricate neural circuits or networks. And it is thought that in the prodromal stage of Alzheimer's disease, amyloid fibrils start to form, and this may impair particularly interneuron synaptic property and function, and start to cause problem in neural circuit function. And also at this stage, it may also start to trigger inflammatory response. And then in the Alzheimer's stage, you see amyloid plaques and neurofibrillary tangle formation, and it is thought that deficits in intracellular trafficking, endosome, lysosomal trafficking, and autophagy further render dysfunction of neurons. And at this stage, loss of inhibitory tone also can lead to hyperexcitability of the system and further destruction of the circuits. So if you look at the landscape of the therapeutic intervention in the disease, so far, all the FDA-approved drugs, they either cholinergic system enhancer or AMDA receptor blocker, they obviously all affect circuit function, specific circuit function, and specific neuronal types. And recently, there's also promising results coming from direct stimulation of the circuits, such as using transcranial magnetic stimulation or deep brain stimulation. So all of these observations really motivate us to think about neuronal population dynamics and how is that affected in Alzheimer's disease. So I would like to introduce to you something, to me, I think is quite fascinating, which is the fact that large number of connected neurons in the brain can fire together to produce this rhythmic oscillation activity. So there are many different frequencies of oscillations detected in the brain. They range from very low frequencies, such as the alpha, the delta waves, to higher frequencies, such as theta and gamma, all the way to very high frequency oscillations. And these different frequencies of oscillations turn out to be highly evolutionarily conserved. You see the same frequencies in very small mammals, such as bat and mouse, all the way to primates, including humans. So this suggests that these different frequencies of oscillation really play a very fundamental role in brain function. So among the different oscillations, gamma oscillations, which are the frequencies ranging between 30 to 80 hertz, are known to be particularly implicated in higher order brain functions like working memory, sensory perception, spatial navigation, and so on. And this gamma oscillations is believed to be the result of activation of local circuits of excitatory and fast spiking inhibitory interneurons. And it is very striking that neuronal spiking in the hippocampus and certain areas of the cortex is known to be phase-locked to this oscillation. So here, for example, gamma oscillation at its peak really prevents neuronal spiking. And the spiking is only permitted during the decay of the oscillation. So this kind of activity results in a very sharp spiking pattern of neurons in these various brain regions. And it is also known that different frequencies of oscillations, such as theta oscillation and gamma oscillations, can be coupled during specific behavior state. And this is thought to really regulate spike timing. So it has been reported that in several models, animal models of Alzheimer's, and in human Alzheimer's patients, gamma oscillations are impaired. So just give you a couple of examples. I don't think that I show all the publications in the literature. So recently, Yadong Huang's group showed that in the ApoE4 knock-in mouse model, gamma oscillations during sharp wave ripples is impaired. And Leonard Milke and Jorge Pavlov's group also show that gamma oscillations are impaired in their amyloid transgenic mouse model J20. And in human patients, it has also been shown that in general, the low gamma power is reduced in human subjects with Alzheimer's disease compared to age-matched controls. So these observations got our interest to ask really how early these gamma oscillations are impaired in the course of the disease. Because if it happens early, it may contribute to the pathogenesis of the disease. So, we collaborated with several of our colleagues at MIT. So my former graduate student, Hannah Alcarino, she works together with Annabelle Singer, who is a former postdoctoral fellow at Boyden's laboratory, and Emery Brown also really facilitated this project. So, we initially started off with a very well-established Alzheimer's model, an amyloid model known as the 5XFAD transgenic animals. So these animals develop a very aggressive amyloid plaque pathology in the cortex, and then eventually, by 6 months of age, you start to see plaque deposition in the hippocampus as well. And by 5 to 6 months of age, these animals also start to show behavior deficits. So we decided to determine the integrity of these oscillations in the hippocampus of young 5XFAD model, 3 months of age, before the behavioral symptom appear and before overt plaque pathology develops in the hippocampus. So, we trained the mouse to run on an air-supported ball through a virtual reality maze. So then we stick the electrode into the hippocampus of the mouse, so we can measure gamma when the mouse runs, and also we can measure gamma when the mouse rests on this behavior paradigm. So here the mouse rests. Okay, so what we found is that we can see this gamma oscillations when the mouse was running, and this is the theta, and when the mouse rests, this is the gamma, underneath the sharp wave ripples. So to cut the long story very short, we found that gamma oscillations do not show significant difference between wild type and the young 5XFAD during the running phase. However, during resting, here, you can see the increased sharp wave ripples in the wild-type animals, and this is the gamma oscillation. This is an enlarged power spectra to show you. And we found that the frequency really formed this unimodal distribution centered around 40 hertz. And what we found is that the gamma oscillations during sharper in the young 5-7 animals is significantly reduced. And neuronal spiking, although it's still phase modulated, the depth of the modulation is also reduced in the 5-7 animals. So, so this is interesting because this mouse was young, as I showed you earlier, and they really don't have behavior deficits. So what does this really mean? So we decided to ask, what if we just introduce gamma into the hippocampus of this Alzheimer's model and see what happens. So we previously collaborated with Chris Moore and Kyle Dicer-Ross and showed that if we use optogenetics to drive the pop albumin positive interneurons, then we can significantly increase the gamma oscillation power in the cortex. And here, if we use the 40 hertz light pulses to treat the animals, then we see this very dramatic increase in the 40 hertz LFP power. And this is very specific to driving the PV neurons, because if we drive the excitatory neurons, we really don't see a similar increase in gamma oscillations. So we use the same approach to drive the activation of the PV neurons in the PV-Cre cross to the 5-FAD 3-month-old young mice. And we see that when we use the 40 hertz pulses to stimulate, we see a very similar increases in the gamma LFP power. And this is very specific to the 40 hertz stimulation. If we use a random frequency stimulation, we don't see such a dramatic increase in the LFP 40 hertz power. So then we wanted to know what happens. And this is when the results became very surprising. So, so we found that if we just perform this optogenetic simulation for 1 hour in this transgenic animals, and then we harvested the hippocampi, and then perform biochemical measurements of beta amyloid levels, we found a very significant reduction of both A-beta peptide 1 to 40 and 1 to 42 following this 1 hour of 40 hertz stimulation. And this is very specific to 40 hertz. If we use more a theta range frequency to stimulate, we do not see a similar reduction of beta amyloid levels. And random stimulation definitely doesn't do anything, actually increase A-beta. And also when we drive KMK2 neurons, even with 40 hertz, we don't see a reduction of amyloid. So we say the data is very surprising, and if we have some idea why is amyloid reduced, maybe we'll believe this data a little better. So, so we wanted to know how is A-beta reduced. So we know that the steady state A-beta levels in the brain is the outcome of how much A-beta is produced versus how much A-beta is removed from the brain. So we first look at A-beta production. We know that the APP precursor protein is sequentially cleaved by beta secretase and gamma secretase to produce amyloid. So following 1 hour of 40 hertz stimulation, we found that both the APP N-terminal beta secretase cleavage product and the C-terminal products, they were both reduced, significantly reduced, and this helps. And also, we found that this enlarged early endosome, which is believed to be one of the subcellular factories where APP cleavage takes place, while this is very prominent in the hippocampal neurons of these 5-FAD animals, following 40 hertz stimulation, these endosomes are dramatically reduced. So further suggesting that the cleavage of APP by its enzymes is reduced. But what's even more surprising came from just some sort of fishing experiment. So we decided to just do RNA sequencing of the hippocampal tissues after gamma stimulation and see whether we can see something different in terms of gene expression. So, so here we found that there indeed hundreds of genes show significant differential expression. But among the upregulated genes, really the surprising finding is that about 35% of these upregulated genes are known to be highly enriched in microglia. Okay, so even though gamma is a primarily neuronal phenomenon, turned out that microglia show the most robust difference in gene expression. And among those genes that are upregulated in microglia, they are known to be involved in phagocytosis, proliferation, chemotaxis, and so on. So microglia, they're glial cells. They are known to support neuronal and synaptic function, and they are also known to play a role in beta amyloid uptake and possibly clearance. But during neuronal insult or neurodegeneration, these microglia are also known to turn inflammatory and can become detrimental. So we decided to take a look of how the microglia look following the 40 Hz simulation. And this is when we see something really very dramatic. So we use microglia marker IBA1 to label microglia in the hippocampus. And so in the control animals, this is what the microglia look like. But following 40 Hz simulation, they simply appear to be completely different. So we try to quantify the differences. Turned out that there are more microglia now in the hippocampal subregions. The cell body basically expand by about 100%. And also, most interestingly, we perform co-immunostaining of oligomeric A beta and the microglia, and we found that those microglia that's positive for A beta in the cell body is increased dramatically. So from about 35% in the control animals to about 90% in the stimulated animals. So this result seems to suggest that microglia become more phagocytic to take up amyloid into its cell body. So far I show you that gamma oscillations are compromised in young pre-symptomatic Alzheimer's models, and driving gamma using optogenetics in the hippocampus markedly reduced amyloid peptide levels in these mice, and driving gamma reduces amyloid production and increases microglia uptake. So when we presented this result to our colleagues at the Picard Institute, they were very excited, but they say, optogenetics is very invasive and it's highly unlikely to be of very high translational value. Maybe you can think of some method to induce gamma oscillations using a non-invasive paradigm. So we went back and a lot of brainstorm sessions with Emory Brown, and we eventually sort of realized that there is some literature suggesting that sensory stimulation is probable to induce gamma oscillations, so oscillations in the brain in general. So the literature I referred to was first published by Wolfsinger and colleague in the late 90s. They shine, or they flicker lights to cats. And then they perform LFP recordings in the visual cortex of the cat, and they saw this very amazing results in that they found that, so this is a power spectrogram. You can see that the kind of frequency they use to simulate these animals, they can record exactly the same frequency in the LFP recording. So this is the plot. You can see that there is a linear relationship between stimulating frequencies and the recording frequencies. And recently, another paper was further published showing very similar results in human subjects. So Hannah Alcarino decided to try whether this approach may work in this little mouse, even though the mouse is known to not have very good visual function. But she used the first generation of this gamma light box. She used Arduino motherboard and some programming to drive the LED light strips to flicker at different frequencies. And then she surrounds, say, a mouse box with this LED light strip, and then house the mouse in the box. And then in the subsequent experiment, we did the LFP recordings in the visual cortex of the mouse, and sure enough, we found that the 40 hertz flicker can drive a very significant increase in the 40 hertz LFP in these animals. Okay. What about amyloid levels? And we found that very similar. So we did a 1 hour, initially did a 1 hour light flicker exposure, and then dissected the visual cortices from the brain, and then we measured amyloid, and we found that this 40 hertz flicker, again, very profoundly reduced amyloid, about 50%, A beta 40 and 42. Compared to the control conditions, and higher frequencies such as 80 hertz or lower frequencies such as 20 hertz don't really reduce amyloid. And if we treated these animals with a very low dose of picrotoxin, the beneficial effect of this 40 hertz light flicker also goes away, strongly indicating that interneurons mediate this effect. And similarly, like the optogenetic-driven gamma in the brain, sensory stimulation-driven gamma also dramatically changed the morphology of microglia and increased colocalization of A beta and microglia. So with these results, we further did a time course to look at how long this amyloid reduction effect with 1 hour of light flicker treatment can last. And it turned out that it lasts for about 12 hours. And after that, A beta starts to go back to its basal level. And by 24 hours, it's completely the same as the pretreated levels. So we decided to do a more chronic kind of treatment. So we did a 1 hour of light flicker exposure per day for a total of 7 days using an older 5-7D mice, 6 months old, where plaque deposition are pretty prominent in many parts of the brain. And we found that this 7 days of gamma flicker treatment can actually remove amyloid plaques in the visual cortex. We found that the plaque number and plaque size are both very dramatically reduced after 1 week of light flicker exposure. And finally, we also use a different transgenic mouse model, the tau P01S tauopathy model, and did a similar 7-day, 1 hour per day gamma Intrument treatment. And then we look at various phospho-tau epitopes in the visual cortex, and we found that this Intrument can also significantly reduce tau hyperphosphorylation and abnormal deposition in the cell body on several different phosphoepitopes. So in summary, I show you that using gamma Intrument using sensory stimulus can activate local inhibitory and excitatory macro circuits to produce gamma oscillations, and this somehow can recruit responses from different cell types in the brain, including neuronal response and microglia response, to reduce Alzheimer's disease pathology. So in just 1 minute, I would like to share with you some of the unpublished results, because every time I gave this talk, people immediately ask me, does this gamma Intrument affects behavior? So I would like to share with you some of the unpublished results. We look at the behavior of the mice with very chronic gamma treatment, anywhere from 1 week to 6 weeks of Intrument. And we found that even in the wild-type mice, young 3-month-old wild-type mice, first of all, we found that these mice show less anxiety levels. So this is shown in using the open field. You can see that this mice spend a lot more time in the center of the open field. And in the elevated plasmates, the treated mice spend a lot more time in the open arm as opposed to the closed arm. And then using Moore's water maze, we also found that although the escape latency during the training period don't seem to be different between untreated and treated, but upon probe trial, these animals clearly show enhanced memory, spatial memory. And in very old mice, such as 17 months old, following 5 to 6 weeks of entrainment, we also see a similar reduced anxiety level and enhanced spatial memory. And furthermore, in the tau P01S transgenic model and the 5-FAD transgenic model, using the Moore's water maze paradigm, we showed that with about 3 weeks of treatment, both mouse strains show very significant improvement in spatial memory. So with this, I would like to close, but just tell you that right now we are testing other sensory modality stimulation and see whether that can also produce beneficial effects in animals, such as this auditory gamma that we are doing in the laboratory to treat animals. And actually, I know that some of the lab members actually also listen to this every day. Okay, so I would like to thank the people who've done the work. Hannah Alcarino, my former graduate student, and currently China Aldaikan, a postdoctoral fellow in the lab, and Anthony Motorell, another graduate student. They're working very actively on this project. And I'd like to thank my collaborators, Ed Boyden, Annabelle Singer, Ho-Jun Suk, who is a graduate student in Ed's lab, Emery Brown, Earl Miller, and Kwang Hong Chan, as well as all my grant support agencies. And I thank you for your attention.
One question there, and then one question here.
Hi, Li-Hue. Hey, Lawrence. That's fine. Here,
Please say who you are.
Hi, I'm Laurie Rajendran from the University of Zurich, Switzerland.
Okay.
Hi, Li-Hue. That was an excellent talk, and I know your paper. And so, I mean, the fact that the gamma could enhance microglial phagocytosis this is absolutely new, and it's also exciting. And one could explain why there is A beta reduction, because the phagocytosis through microglia is enhanced. But what I'm surprised is the improvement in the behavior, because my lab just recently published that enhancing microglial phagocytosis would reduce A beta peptides, but at the same time, it would also enhance synaptic pruning and synapse removal. So how do you explain your reduction in A beta, but at the same time, improvement in the memory?
Yeah, so I think as I try to allude to, I think this system recruits many different cell types, many different pathways activation. So microglia activation is really just part of the story. Neurons really, I mean, both the interneurons and excitatory neurons, they undergo dramatic changes as well. So we recently have done a lot of omic analysis, proteomics and RNA sequencing, and we clearly can see the synaptic components really undergo dramatic changes. Astrocytes, the vasculature, they all undergo dramatic changes. So I think, we're just trying to figure out, really try to tease out, which function, how can we, decipher the underlying mechanisms.
Have you checked in these
So
microglia?
in fact, in all the Alzheimer's models, their reduced synaptic density, they are all completely ameliorated. So synaptic density is definitely increased.
Yeah. Hi, this is Rafael Fernandez Chacon from Civil. Hi, very impressive, Li-Hue, I really like it. So the question is, so what do you think is going to happen if you do the opposite thing, if you remove or you silence gamma oscillations?
So you mean the inhibitory?
Exactly.
Yeah,
if you use halorhodopsin, for example.
I would speculate that it's probably gonna exacerbate the pathology. That would be my speculation, but the
But
experiments
in a
have to
wild
be done.
type, in a wild type, what do you expect in a wild type?
In a wild type, it has been shown, right? I mean, loss of inhibitory excitatory balance can have, a lot of psychiatric and, epileptic form, kind of a lot of detrimental consequences. Hi.
Brian Strange from Madrid, Fundación 100. Again, I mean, I'm very impressed with the paper. It really is extraordinary findings. Would you make a comment about endogenous gamma, and particularly in the human? I mean, we're in the state of gamma very frequently. Do you think being in endogenous gamma might be recruiting the same mechanism of microglial activation?
I think so. But endogenous gamma usually is very transient. Okay, so the gamma, it will be activated and then it goes down right away. I think here we are just artificially really prolong gamma oscillations.
Although in some situations during cognitive tasks, I mean, we see gamma evoked by stimuli that can be quite sustained.
Which what?
The gamma activity can be sustained if you put a person in the context of a task.
Yeah.
So it's
I think that it would be very interesting to figure out. I'm actually collaborating with Alvaro Pasqualeone. Are you here? So we are about to start to do some experiments in human subjects to look at this more closely.
Thank you so much. We can
Thank
ask
you.
further questions in the final bit. Li-Hue?